Hi Alex, how are you? Oh, how did your mock go? Did you do it? Hi Chidima, how are you? I'm hoping that it's not going to lag tonight as much as it did the other night. Not great. Oh no. Wait, I didn't hear what you said. Sorry, please. I was asking Alex how your mock went, is what I was asking. Why is it not great? Is it because year 13 started as it means to go on and it's now too stressful? Yeah, I bet it is. That's the problem, year 13. It literally starts and then you just don't stop, and then it's the exams. And it's trying to get on top of things now, as you mean. I know, Friday, isn't it? I don't have any plans. That's all right. Oh, I'm glad, Alex, that it went okay for you. That's good. Guys, what would you like me to look at? The timing was diabolical. Why? Do you think exam practice is more important than learning the content? Oh, I think it's a bit of both, to be honest. I wouldn't say that it is one over the other. You need to be one knowing your content, but you also need to do the practice because if you've not done the practice, you're not going to get anywhere. You can memorize the content and then just try and regurgitate an exam, and it won't help you. Um, Biodiversity, OCR. Yeah, Chidima, that's I'm over there as well right now. They gave you 40 minutes for a 60 mark assessment. That's shocking. Liver as well. Okay, so what about memorizing mark schemes? Not good. Don't memorize mark schemes. Unfortunately, the mark schemes are only specific to that year's exam and they can change. What's important is knowing the key words that are in the mark schemes that are basically accessible to all the questions relating to that topic area. So yeah, don't memorize mark schemes. Chidima, here you are. Nice to see you. Right, okay. Oh, first of all, can we just like say, well done, Christine? I finally finished my study guide for exchange and transport and animals. I actually have put together the exchange system knowledge and the transport and animals content, and it's like a storytelling um study guide in a way that it should make it so much more accessible and easier to note. So yeah, definitely. Right. Alex, you said the liver. Where have you got up to with the liver? Are they just doing the structure of the liver? Thank you. Do you know what? It's been hanging over my head because the whole summer holiday looking after the girls, I didn't manage to do what I wanted to do for it. Um, so it's been like something that's been hanging over my head. So I'm so pleased that I got it done today. Obviously, the next step is the revision guide. Not the revision guide, the workbook to go alongside it. But that's another lot of work that I will get done at some point. But liver, where did you get to with the liver? Hi. Why do I discover you right after? Oh, I'm sorry, it's right after your A level. I did try, and get everybody to find out that I'm here, and I did so many lives. But where can you access it? ukbiotutor.com is my website where you can access it. And obviously, if you don't know already, I do have my YouTube channel as well, which has got the videos available for you there. So you got to love a bit of urea production. That's easy stuff, I would say, in detoxification. Oh, nice, Alex. Well done. Oh, thank you, Chloe. Thank you. Okay, let's do a bit of the liver and then I will jump across to the biodiversity. Chidima, I know that you love a bit of respiration, but it will be coming. Oh, the endotherms and ectotherms, actually. We'll look at that. Do that. Oh. Hey, there's no issue with applying to other places and also still applying to Cambridge. It's important that you do what's needed to get you where you need to go. You should be proud of yourself. You sat it. That's the key thing to note. You did actually sit it and you've put the effort in. Right, we're going to look at the liver then. Alex has asked for liver, so we're going to start with the liver. It's important to remember, this is one of the key things that's come through time and time again, that any of the organ systems that we're going to look at, you need to make sure that you note that they have got arteries which are leading to capillaries, which are leading to veins. Now, let me actually open up my booklet and I'll add in a diagram here as well. It comes straight from the workbook. Study guides. Just bring this down. So, thank you for the likes, Riley. Thank you. I'm just trying to show this at the same time. Okay, so the one thing I would say to you, so I'll give you a little sneak peek of the workbook. I've gone too far, haven't I? Yeah, this is what I want. So when you are looking at the blood vessels, this was something that a lot of students got caught out on this year, in the fact that what you have is arteries which lead to arterioles, which lead to capillaries, capillaries which lead to venules, venules which lead to veins. Now, if we're looking at this diagram, my little note that's beside it, you'll see that it says an artery to a capillary to a vein. But whenever we talk about tissue fluid formation, so if I scroll down now to the tissue fluid formation section, whenever we're looking at the tissue fluid formation, we always talk about the arterial end of a capillary, and then we talk about the venule end of a capillary. So it's really important when you're thinking about all your organ systems that you remember that the arteries are leading to the capillaries, but there are arterioles before we get to the capillary, and there are venules before you get to the vein coming from the end of a capillary. So when we look at the hepatic vein, which is going to take the deoxygenated blood away from the liver cells, because it's a hepatic vein, that's something that we need to take into consideration. And then the other thing we need to take into consideration is where our hepatic artery is as well. So the hepatic artery is going to be leading into the liver. It's going to be leading to the capillaries within the liver, and it's going to be carrying that oxygenated blood to the liver. The hepatic vein is going to be taking that away, that deoxygenated blood away from the liver. So there, that is actually just looking at your module 2 knowledge. No, it's not module 3 knowledge, sorry. Looking at, do you know the blood vessels and do you know how they are connected? So you've got your arteries, you've got your vein, and they are linking together. Let me just that and to just have this one. Don't know if you guys can hear the rain outside. It's proper pouring down now. Okay, so if we are looking at this hepatic artery, we're looking at this hepatic vein, and we're talking about the blood going in, we should therefore remind ourselves that our capillary is the place where we are going to have the exchange occurring. And that's what we'll be looking at. Yes, I will also try and bring in a bit of the hormonal response because it links quite nicely to the liver topic area. So the other one we have to know about is called the hepatic portal vein. And the hepatic portal vein is going to bring products in the blood which have been absorbed from the intestines. So we're going straight from the intestines. So all of those digested molecules that have now been absorbed into the bloodstream will be carried to the liver via the hepatic portal vein. What we've also got is we've also got our pancreas that's going to be secreting hormones, linking to the hormonal system. It's going to be secreting the hormones, and therefore, when those hormones are being secreted into the blood, that's also going to be carried to the liver via the hepatic portal vein. So this is a portal vein because it's coming from another organ, and it's making its way into the liver. So therefore it's the hepatic portal vein. So it's got that blood, which has got lots of the products from the digestion that's been absorbed from the intestines into the bloodstream. We've got hormones, which are being secreted from the pancreas, and also it is a delivery system from the spleen as well. And your spleen's involved in the breakdown and the immune response as well. So there's lots of molecules which are being transported in the bloodstream, and they're making their way to the liver. Now, another thing for us to note when we look at this is, 1 second. Oh, almost accepted to do a live with other people. That's not what I meant to do. The other thing for you to know is the hepatic portal vein is delivering this blood to the liver, but the liver also has this extra bit, which is the gallbladder. Now, the gallbladder is important, especially if you are doing AQA. Obviously, we're doing mostly OCR, but if you're doing AQA, you need to know more about the gallbladder in the fact that that's where bile is stored and the actions of bile. For OCR, you just need to know about that storage in the gallbladder of the bile. So 1 second, let me just get rid of those bits above. This is where I'm trying to bring my notes from previous lessons. Makes it a little bit more tricky now. So you should have looked at the lobules that you get, Alex, in the kidney. So when you're looking at the liver, you need to understand that it's actually divided up into these lobules, which are these rows. They have these rows of hepatocytes. Now, hepatocyte is basically, a cyte is a cell. So a hepatocyte is a liver cell. So you've got these rows of liver cells, and they're arranged in this circular pattern around a central vein. And what you end up getting is, just bring this in, this area here where you have these little sections leading down. Now, the histology is probably one of the hardest parts when it comes to the liver. They do expect you to know the histology of the liver, and they, yes, well done. The hepatocytes are liver cells. Absolutely right. And so they do expect you to know, oh, 1 second, click the wrong button now, the structure, and it came up in 2023's paper, no, 2024's paper, where you actually had to identify the different arteries and veins leading to a picture that looked like this. So it's important whenever you are looking at the liver and the lobules, you need to understand that there is a hepatic artery, a hepatic portal vein, which is going to connect with that hepatic vein, which leads out. And the way that they connect, oh, Iman, that's amazing. You got into UCL. Wow, well done. actually, it's not down to me, it's down to you. It's down to your hard work. So well done. That's awesome. Coming back to the liver, the key thing about the liver and this connection is knowing that you have got what's known as a sinusoid. There we go. That's Alex's goal. Alex's goal is to get to UCL. There's your sights are there. These people have just sat their exam and they've got what they needed because they worked for it. And that's exactly what you've been doing, Alex. So keep going with that. Right, back to this. Hepatic portal vein, hepatic artery. Remember what I said about the transport in animals topic area? The transport in animals topic area is important because you should know and be able to identify the lumen size to identify whether you're looking at a hepatic artery or whether you're looking at a hepatic portal vein. So now that they have actually published the papers, it means I can show you the question that I'm talking about. Give me 1 second whilst I get it up. Whilst I'm doing this, whilst I'm getting this picture up, you guys can maybe answer me a question. I'm considering whether or not to start a biology study club Discord where you could join the Discord and ask questions relating to, there we go, this is the one I want, the topics that you're struggling with the most. What do people think on that one? So let me know, put a comment so I can help you. That's not what I want. You can go away. This is the one we want. And this is the question that I was referring to. So you have your, let me put that, go up a second. There we go. So here's the question, and you're told in this question that you have got a liver tissue. So you're already being told that it's liver tissue that you're looking at. Yeah, I know. This is why I'm trying to figure out if Discord is the right place to do it. I'm trying to figure out what the right thing is to do as a location where you can come and write questions and be part of a community, which is the OCR community. But I'm still figuring it out. I wasn't sure as well. That's something that I've is on my con list. So here we have stain sections of a liver tissue, and they want to know. Okay, so you're saying you think it's either B or D, but probs B. So Alex, my question would therefore be, why is it not A and D? A and C, sorry. You said it's either B or D. Why is it not, do you think, A or C? Because whenever you do these practice questions, you need to be able to eliminate the wrong answers. And if you can't eliminate the wrong answers, it means you don't know the topic area enough. So why is it not a hepatic artery? Why is it not the hepatic vein? I have not learned the liver yet, so I have no clue. It's coming. Probably one of the harder topic areas that most students struggle with aren't the largest ones. So, okay, so this one here, if they were pointing at that one there, that would be the hepatic artery. So it would be the hepatic artery. Yeah, some students do love the liver. I love the liver. It's interesting. It's like really you get into nitty gritty part of it, but others don't. But yeah, so this is the hepatic artery because it has a small lumen. So we can tell that it's not the hepatic artery because the lumen is much bigger. So we can eliminate A for that reason. but can we eliminate C? So you said it's not the largest, and that would be, this one here would be the hepatic vein. So if I was to eliminate that one, so now you need to identify, well, what's a sinusoid and why therefore, because you said it'd probably be, it is B, it's not the sinusoid, and why is it not the sinusoid? So the sinusoid is the specialized capillary in the liver lobules, and these are actually these parts here that you can see running down. It's not very clear in this picture because it looks like it is a grayed out version. Most of them you will see that it's clear. That's right. So you're looking at the ones that are leading down to that central vein. They are your sinusoids. Good. Okay, so if we look then at the hepatic artery. Oh, no, don't want to do that. Give me a second. I need to move this one down slightly. Again. Move that one down. There we go. So what we have is blood will flow from the hepatic portal vein, from the hepatic artery, and it will flow down through the sinusoid. So the sinusoid is the connecting capillary from that hepatic portal vein, from that hepatic artery, and therefore the blood will flow down into the hepatic central vein. So what we need to understand is, well, whilst the blood is flowing down through the sinusoid to the hepatic central vein, is a sinusoid not a sine wave? What do you mean by, Ben, what do you mean by that? What do you mean by a sine wave? So what you have is that blood flow is going to flow down through the sinusoid. And what you can see is surrounding the sinusoid are the hepatocytes. Now, your hepatocytes are your liver cells. And your hepatocytes are multipotent. So this is going to bring in your module 2. I don't know. It's going to bring in your module 2 contents. This is 2.6. Do you understand that hepatocytes are multipotent because they can divide and differentiate? So your liver is the place that can regenerate itself if it has damage, because it is dealing with detoxifying the blood and the substances within the blood. It is important that hepatocytes have this ability to divide and differentiate, to replace and repair damage that has occurred within the liver. Now, not only are the hepatocytes multipotent, so therefore can divide and differentiate, when they do differentiate, one of the identifying features is that they have these large nuclei and prominent Golgi apparatus. So what we're saying is that they are metabolically active. They have got these large nuclei so that they are able to store that genetic material. Excuse me. What they also have is prominent Golgi apparatus. These prominent Golgi apparatus, remember, Golgi apparatus is about modifying and packaging the proteins within the cell. And the fact that it's got lots of mitochondria tells you that it's going to be producing lots of ATP. And so it's going to be doing aerobic respiration. So this is where you need to link together all your different topic areas. So we've got aerobic respiration occurring. For those of you who are doing respiration, you should know that it's aerobic respiration because the mitochondria is where you have your Lynx reaction, your Krebs cycle, and oxidative phosphorylation. So the fact that the mitochondria are there, there's lots of them, that is for aerobic respiration. because anaerobic respiration happens within the cytoplasm. So this is where they could say, right, we're going to ask questions on the liver, but we're also going to assess your respiration knowledge at the same time. So do make sure that you are interlinking those topic areas together and not just thinking that it's one or the other. It's not. Biology is this bigger picture, and you have to be aware of all of it. Okay, so we have got the hepatocytes, the fact that they're multipotent. We've got that they have differentiated after they've divided by mitosis, genetically identical daughter cells being produced. They've differentiated and they therefore will do their role. You are very welcome, Alex. The other thing to note as well is you have got specific types of macrophages. So remember phagocytes. This is module 4 coming in. You've got these phagocytes. They are macrophages, but they've got specific names for the ones which are found and located in your liver cells. So these are called Kupffer cells. So the Kupffer cells are just a specific type of macrophage which is found inside the liver sinusoids. And therefore that is needed for engulfing and digesting debris that is going to be circulating. Oh, it's not very clear because of the position. Let me move that 1 second. The other thing that you need to also note is you have what's known as your bile duct up here. And the bile duct is where bile is going to be released into. So bile is actually made by hepatocytes and it is secreted into the bile duct for it to then be transported down where it's going to be stored in the gallbladder. So let's look at bile production then, and let's see what actually happens when it comes to the production of bile. So we know we've got our hepatic portal vein, our hepatic artery, we've got our sinusoid down to our hepatic central vein. What we have to understand is that our hepatocytes are going to be producing bile. Now, to do that, you have to take a step back and remind yourself about erythrocytes. Module 2 coming in, erythrocytes are a differentiated type of cell that you should know about. So module 2, differentiation. Module 4, phagocytosis. Erythrocytes, when they have differentiated, that's absolutely right, Alex, they have no nucleus. Well, before they got rid of the nucleus, they had a nucleus, because remember, the erythrocytes are differentiated. They had to come from a stem cell that went through the process of mitosis. So the hemopoietic stem cells that are going to go through mitotic division to produce the erythrocyte is then going to produce lots of proteins. It doesn't get rid of the nucleus until it has made as many of the proteins as it requires. What are those proteins that are found inside the erythrocyte that were made before the erythrocyte got rid of the nucleus and all the other organelles? So that would be a question I want to know. Hi, Chris. I did have a lovely summer, thank you. I did. I was in the south of France with the girls and Alan, and we had some glorious weather. It was really nice to get away. Alex, no, it's not histones. It's not histones. So the histone proteins are associated with your DNA. What I'm really interested in is what proteins have been produced within the erythrocyte. And this is one that so many students don't put together. You learn module 3 and you learn all about the blood. You learn all about, thanks, Chris, for the likes. You learn all about the fact that the blood's role is to transport something, but to be able to transport something, the erythrocyte must have this globular protein inside it, and it has millions of it. It has actually 300 million of these molecules inside the cell because it's cell. Yes, hemoglobin. So the erythrocytes have made lots of hemoglobin. They've then differentiated, got rid of the nucleus. They have got rid of any organelles they don't need, and they've got this biconcave shape so that they can obviously move around and squeeze through those capillaries, the sinusoids. Every capillary that is within the body system. And these are really small lumens in the capillaries so that they are ensuring there is a short diffusion distance. So why am I going through all this content about erythrocytes and reminding you about that? Amna, how are you? Good to see you. I'm good, thank you. I am very good. How are you? How's uni going? Anyway, back to this whilst I was answering that question. So the erythrocyte, the reason I'm talking about this is the erythrocyte doesn't have an unlimited lifespan. It's got a very short lifespan. It's got 120 days that it can last before it no longer functions. So the Kupffer cells are going to engulf and digest the erythrocytes that are no longer functioning. And so what the Kupffer cells do is when they break down the debris from these erythrocytes, they are going to digest hemoglobin down. And what happens is, oh, nice. Oh, lucky you, not started yet. They're going to digest the hemoglobin down into the heme group, the prosthetic group, which is part of the hemoglobin and the globular protein. Hi, Alex. So they have the heme group, which is your prosthetic group, which has your iron core. Now, remember, the iron allows the binding of oxygen in hemoglobin. The globular protein, the globular part of it is because it is a 3D shape. It's also soluble. Thank you, Alex. Also soluble. So we have this hemoglobin, this globular protein, which is soluble. A lot of students forget to use that in their exam. Soluble protein, which is dissolved in the cytoplasm of the erythrocyte, and the hemoglobin is binding the oxygen for oxygen transport. So when the erythrocytes no longer able to carry out that function, it needs to be broken down, it needs to be digested, and that is where the Kupffer cell comes in. So it's going to engulf, it's going to digest it, it's then going to end up with the heme group and the globular protein, and that globular protein can be broken down into the amino acids, and the heme group can be split up into the iron and bilirubin, which is bile's pigment. So the iron is going to be taken back up to make more erythrocytes, but the bilirubin is needed to produce bile. And so the hepatocytes are going to use the bilirubin that's come from the breakdown of the heme group to produce bile, which will then be secreted into the bile duct. Oh, thank you, Amna. Thank you. And the iron obviously needs to be taken up because in the bone marrow, we've got our hemopoietic stem cells that are going to be producing more cells through mitosis and then the differentiation into erythrocytes, and erythrocytes need the iron to make the hemoglobin. So it's kind of like a way in which the body can ensure that it is able to transport oxygen around efficiently by not wasting that iron. Oh, nice. I'm glad you're finding it easier. I'm trying to look, you're writing on both sides, Alex. Okay, so remember, I talked about the gallbladder before. That bile is released into the bile duct within the liver. It is then transported down to be stored in the gallbladder, and then it's released into the small intestine. And the bile pigment is actually the bit that colors your feces. So you can tell if there is issues with your bile duct, if there is problems with, for example, the color of your feces. Also, it's linked to bile is necessary for your fat digestion. And so therefore, the bile salts that are released are also necessary for emulsifying the lipid so that lipase can get access to it. So all of this is interlinked, but what they tend to do with these types of questions, when it comes to bioproduction, they give you little bits of information, but what they want you to understand is how to apply that to, for example, an exam question. I'll throw an exam question up now, actually, that they did a number, I want to say it was 2022's paper, where they threw a question up, which was a graph. And in that graph, they wanted students to, there we go. I just saw it a minute ago and now I've lost it. Where did you go? There we go. It was talking about problems with the liver. And so they wanted you to know about psoriasis. They wanted you to know about different proteins. But proteins that you'd never heard of before. And this is the bit that a lot of students panic with when they get an unknown situation that they've never come across before, and they are sitting there going, I've memorized the bioproduction. I've seen the video that Christine did on the bioproduction, but this question isn't about the bioproduction. This question is about a protein that I've never heard of before. So you're told in the question that the cirrhosis of the liver can result from long-term liver damage. You're told that there's alcohol or other toxins can cause this damage. And they've suggested that it can be detected by these two proteins. So they've given you some bits of information. They've told you you've got your C-reactive protein and you've got your copeptin. So they've shown in the key in the graph that you have these different proteins. So now what they want you to do is they want you to understand what they're showing you. So at the bottom, you're told you've got blood, you've got saliva, you've got urine, and you've got the feces. So they have measured the concentration of these two proteins, and they have looked at it in 4 different locations. So how does that, why is there much more C-reactive in the blood? Why would you expect that to be the case? It is what it's showing you in the graph. But why would there be more of this C-reactive protein in the blood? No, it's not due to the hemoglobin levels. Because hemoglobin levels, remember, every erythrocyte has about 300 million hemoglobin molecules. So we're talking about this showing you that there is damage in the liver. So why would there be this protein in the liver being released, indicating liver damage, but be high in your blood? Because it's not high in saliva, it's not high in urine, and it's not high in the feces. So the C-reactive protein is high in the blood. Why? How does that indicate anything to do with the liver? What do we know about the liver? Well, we just spoke about the liver and the fact that the liver has got these lobules, right? So we have these lobules, lots and lots of lobules, which are being connected by our sinusoid, by our hepatic portal vein, and our hepatic artery. And this is our central vein. So we have loads of them because the liver has a large blood supply. Therefore, if there is a large concentration of these C-reactive proteins in the blood, that's because where does the liver damage get detected first? That release of those substances is straight into the bloodstream. So we would expect there to be a higher concentration within the blood because there is a large blood supply to the liver. So that's the first thing that it's looking at is, do you actually understand the structure of the liver? Do you understand that there's these many lobules making up the liver? The liver is like the largest organ apart from your skin in the body. It's the heaviest as well. So that would explain why we have this high concentration in the blood of the C-reactive protein. Very good question. As bile pigments have an inclusion in feces, you're absolutely right. But what can you see in the graph? What can we see in the graph that makes sense to explain about what I just mentioned? Bile being a product of the breakdown of the hemoglobin to produce the bilirubin being used to produce the feces. What in the graph shows us that the bile that's produced in the liver contains a product showing that there is a breakdown within the liver. So Alex, if you're looking at the graph, what part of the graph is showing that? Not a problem. This is one of the hardest questions that was ever asked when it came to the liver and an application question. So I'm saying you asked me about the fact that the bile, so you said on here, why not feces? As don't bile pigments have an inclusion in feces? So feces doesn't have a high concentration of the C-reactive protein, but what does it have looking at the graph? Very good. High copeptin levels. So looking at the graph, we can see that there are high copeptin levels compared to the other places. So therefore, that is another way in which we're indicating, because bile is made and colors feces, should say made in the liver, and colors feces, and therefore that is another indication as to why, if we have liver damage, if we're finding large concentrations of this copeptin, that is because the feces has got the bile in it The bile was made from the liver, which has the damage. So it's a real mind-boggling question when you look at it to begin with, because you're like, I've never heard of this C-reactive protein. I've never heard of copeptin. I've got a graph that's showing me these concentrations. How does that have anything to do with liver damage? And that's what they're looking for you to understand. Use that knowledge that you have. Right, we've got large blood supply to the liver because of the sinusoids. We've got this, and we've got the two hepatic artery and the hepatic portal vein leading into every sinusoid. And also we know that the hepatocytes produce bile, which is going to color our feces, and therefore that's why we would find those products there. So it was a question that threw so many students off when they came to sit it in whatever year they sat it. I want to say it was 22. Oh, actually, no, that was 2017. That was way back when they first did the exams, when they first put them out. Oh, what was the actual exam question wording? Hold on 1 second. It was suggest why blood and feces have the highest concentration. So you didn't have to notice it yourself. They wanted you to explain why it did, and it was worth 2 marks. 2 marks. There we go. Yeah, exactly. It wasn't the only question that that one had. That one also threw a lot of students because they were asked to do a calculation and do some math. So let's finish off with a little bit of maths on this one. And then we'll go from there. Okay, so here we go. This was it is an intimidating question, which is why I like to show the hard questions, because I think doing the easy questions sometimes is too easy. And it's about understanding those hard questions. How do you work them out? How is it you get the understanding to improve? It's not just about memorizing mark schemes, it's about understanding why the mark scheme is correct, what it means, and how could they ask the question if they're going to ask it again in a slightly different way. So the content is the same, but they just ask the questions in a slightly different way, which makes it more confusing for some. So this one is looking at calculating the order of magnitude. So what would you do for this one? Is it 9? Why do you think it's 9? Why do you think that it's 9? We're looking at the copeptin in feces. So we need to do feces and they want copeptin. So we therefore need to look at this one here, which is 10 to the 9. And then they also want us to look at the C-reactive protein in saliva. So we've got saliva, we're looking at the C-reactive protein. So we're going across here. Just change that color. Going across here. So therefore we've got a 10 to the 9 and a 10 to the 1. So how would you calculate the order of magnitude? There we go. So you would have 10 to the 9 divided by 10 to the 1, which would give you your 10 to the 8. So many students actually still got the marking point, even though they wrote 10 to the 9 minus 10 to the 1, which would obviously give you 10 to the 8 as well. But although they gave it 1 year, doesn't mean that they would give it another year. So it's important that you work it out and know how to do the order of magnitude. Right, I'm going to finish that for tonight. Thank you to those of you who have joined me. I will be back next week again. I'll be live on a Wednesday at 9 o'clock, as I have been doing. And yeah, definitely wouldn't rely on the wrong method. Oh, Matthew, do you know what, Matthew? Matthew, I'm so glad that you've jumped on here. Thank you so much for buying my study notes. I see your name pop up all the time, and I was thinking about actually emailing you because I think it's lovely that you actually are finding them useful. So thank you for jumping on here and saying that you're finding them really useful. I literally have just released the uh, exchange system and transport an animal. And what I did with this one is slightly different to my previous ones before, because this one I've told the story. I've realized that I'm not playing to my strengths as much as I should be in the fact that I tell the story of how they all interlink together. So I would really love to hear your views on how you think the new one that I've just done goes. So yeah, definitely please do check it out. And do you know what? Once I do a workbook on that one, I do want to give you something for free because you've literally bought every single one of my workbooks, and I do see your name pop up, and I'm like, thank you. You are my like true subscriber. Um, oh, good luck with your exams. How much it depends on which one you're looking for, Chidama. So they're in the Uh, range of it depends because it could be £2.99. It could be the one that I've done today, which I've just launched today, which is all of module 3 exchange system and all of the animal response. That one is £8.99 for the whole lot of information. No, thank you. And I will definitely, once I've got the workbook ready and going, I will send that. Well, this is what I've tried to do. I've tried to keep it as cheap as I can do for you. Oh, Mel, nice to see you. You've helped me so much in my A level last year. Oh, thank you. Mel, were you not the one of the people who got them for free? Did you find them useful? You were one of my winners, weren't you? Yay, there we go. Oh, I'm pleased that you found them. Again, I was going to send some emails because I like feedback. I'd like to know how people find them. Oh, good. I'm glad you found them really helpful. I'm in the process of building the rest as well to keep going. I want to have a full pack of them, but I want them to be the right information to make it not just like a revision guide that I've thrown out that you could buy like any other one. I want it to be something that is useful, that actually helps students and makes it make sense. So I'm really pleased to hear that you find them useful. Um, I was having a good day that day. I was like, oh, send it, put a thing in, and you'll be one of my 5 winners potentially. So, right. Anyway, I need to go to bed because I have got a busy day again tomorrow. And yeah, thanks for joining me. Thank you guys. And I look forward to continue to working with those of you who are year 12 and those of you in year 13. You definitely need to be at my lives because it's going to just get harder and harder. AQA, I tutor AQA privately, but all my resources have mainly been by Amna, have mainly been for OCR because there wasn't enough out there for OCR. I am going to look at what I've got and adapt them to the AQA. The one that I've done today actually is relevant to both. Although it is OCR specific, it's actually relevant to both because you have to do the fish, you have to do the insects, you have to do exchange the ventilation in the mammals, and because the surface area to volume ratio side of things. So that one is very relevant across both exam boards. Oh, you have your first assessed practical. Apple, don't worry about it. The assessed practicals they're not meant to be stressful. What they're meant to do is they are meant to assess that you have gained skills, and you're actually meant to fail some of them anyway. If you're passing every single one, you are not showing improvement. So you're meant to not get approved on some of them because you're meant to be coming and learning skills by doing the practicals. Um, so honestly, don't worry about it. Yeah, PAGs are awesome. They're so much fun. I love doing PAGs. My students hated it because they'd come into the lesson and I'd be like, there you go. They're like, what do we do? What do we do? I'm like, and I had one student actually at one point when we did the dissection of the heart, he actually videoed it and I was like, show me your video. I want to see it. Because I want you to have fun. You should have fun while doing your PAGs, but also you should actually be able to follow instructions. So your teachers need to assess you, but they should also make it fun. There's always an instruction sheet, always, always an instruction sheet. You just got to follow it. Oh, nice. Heart was amazing. And the kidney is hilarious because the kidney stinks. And I'm always like, ready for this one. So yeah, the kidney's a good one because it just becomes like this mush. Wow, did you do Daphne? Were you looking at the heart rate and did you give them caffeine? See, it's a funny one. When I used to teach the IB, before I moved over to do more on the OCR, they were very against animal testing. And so that wouldn't be allowed in the International Baccalaureate. So we didn't do it in the school when I was doing OCR. But I remember I was looking at some uni work the other day there, and I remember when we actually had to inject some crabs and watch what they did. It was crazy. Crazy to think the things that we did, but we're going back, what, how many years now? A good 20, 24, 25 years, not to give my age away. Oh, you actually dissected a locust as well. Oh my gosh, your skills are awesome at the practical side of things. That's really cool. That is really cool. You're very lucky. A lot of skills don't go into that many practical situations. My eyesight now would be horrendous for doing those sorts of practicals. It really wouldn't be good. Um, Biological molecules is really hard to start with, but Apple, you're going to love it because it's going to be the easiest part of the whole course. It is horrible to begin with. Don't get me wrong, it is horrible. So if you're just starting in year 12, it is hard and it feels like it's really rough, but actually it's not at all. It's quite nice. Um, what are my predictions? Interesting. What are my predictions for next year already? I feel like nitrogen cycle's coming up because it didn't come up this year. So basically, what I would tell you to do, go watch my videos on my predictions that I thought were going to come up this year. And anything that didn't come up is coming up next year. So nitrogen cycle, I feel like is definitely going to come up. Um, but I could be wrong because the person hasn't written them yet. So how impressive is an A star in A-level biology? Well, clearly it's impressive because only 9% of the cohort gets it. Was it 9% or was it? I think it was actually lower than that. I want to say it was 8 point something or other. Give me a second. Let me look that one up. 2025. Where are you? Spreadsheet. My other spreadsheet on. I did see Alex. Well done. I did see that. I also saw Chidima and her response to that as well. Um, uh, Do you think you can get an A star in this year's OCR A level? But do I think I could get an A star? Honestly, um, I actually don't know. And that sounds bad because I teach it and I can teach it. I don't know if I could get I can get an A. I can absolutely get an A 100%. Could I get an A star? I would like to think I could, but my problem is that I take a lot longer to process information, and I second guess myself, and I sometimes write not what I'm thinking or not. So when I sat the exam paper like I was a student, so every year when you guys sit the exam papers, when I get access to them, I sit them as if I'm a student to see how I would do under exam conditions. And when I did that, I made some silly mistakes because I was feeling rushed and I was doing things really quickly. So I think because of my ADHD, I would be on the just under potentially. I'd either get it or just be under. Because it's not easy and there are some difficult questions. But does that mean that I don't know the content? Absolutely not. I do know the content. I know this course inside out, but it's just sometimes the way they word questions is confusing. What's the hardest topic in A2? It's an interesting one. It really depends. Different people have different strengths and different people have different weaknesses. Which is the hardest that I think that most students struggle with? The hardest topic I would say is module 2, and it is probably gene control and manipulating genomes. They're probably the two, not module 2, module 6. That's what I meant, module 6. I'm thinking of module 6.2, literally. Yeah. See, and this is the thing when someone said, would I get A's? That's what I do. Sometimes I think something, but I say the opposite. And because I'm saying the opposite, it's not that I don't know it, it's just in my brain it gets a bit muddled up. But I know that about myself now, and I would know that I'd need to slow myself down. Um, but I would like to say that I would get an A star, but I don't know, to be honest. I don't know if I would. I'd get an A probably. You should be absolutely proud of both of you because you have done everything.